Hello everyone, let me introduce you to Nisha, Naspreet, Shamirah and myself Yashu. We are from Group 2 and the title of our journal will be Effective Microorganism to Improve Quality of River Water. Our chosen article was published in year 2016 by Waida Wahid and Shamila Azman. In this article, the effectiveness of effective microorganism is carried out at Sungai Sebelung, Kampung Melayu Majidi, which is located at Johor Bahru, Johor. Moving on, don't we all wish to have clean river water? However, with urbanization comes pollution, the death of aquatic organisms, and less sources of fish for consumption. Our rivers are crying for help. Thankfully, we have our super mud ball to the action. This mud ball consists of numerous effective microorganisms which should be further explained. Now, let's look at the purpose of the study. Firstly, it is due to pollutant impurities that impede adequate supply of clean water have a detrimental effect on the quality and harmful for all living organisms. Next, with increasing trend in population growth and socio-economic development, the quality and quantity of water is gaining widespread attention from all over the world. Thankfully, we have effective microorganism, which is a promised technology because it has no negative effects to all living organisms and the environment. Moreover, it's a low-cost solution too. In Malaysia, effective microorganism is known as EM-activated solution. EM mud ball is widely used in Malaysia, and this is because the mixture of molasses for EMAS is usually from sugarcane and EM in non-chlorinated water or rice rinds. In this journal, effective microorganisms were being chosen as a solution to reduce the water pollution at Sungai Sebelung because no particular substance that can harm the aquatic life is used and the cost to treat the river is low as EM only use waste material. Hence, this method is the suitable method that should be practiced. Now, let's look at what specific questions were addressed and how. First, what are the water quality status of Sungai Sebelung after being treated with effective microorganism mud ball? Next, what is the comparison of water quality data with water quality index and to investigate whether effective microorganisms are effective in the river conservation at Sungai Sebelung? And finally, is what are the factors that causes pollution at Sungai Sebelung? That's all from me. Thank you. Over to you, Naspreet.
Moving on to the methodology. Samples were taken from a total of 6 locations. An in-situ analysis was carried out to obtain the precise pH, temperature and dissolved oxygen reading. To determine the water quality, 5 parameters were tested in the laboratory analysis, which will be shown in the next few slides. The graph shows dissolved oxygen at different sampling stations, whereby based on the graph, it first shows a decrement and then it increases and lastly it decreases again. So based on the National Water Quality Standards (NWQS), the average dissolved oxygen value, which is 3.59 mg per liter, falls under the category of class 3. Biological oxygen demand graph showed an inconsistent trend. The average value of BOD obtained is 18.39 mg per liter and it is classified as class 5 based on NWQS. Previously, the BOD concentration was even higher, which is at 29.3 mg per liter and 20.26 mg per liter. Therefore, the usage of effective microorganisms can improve the water quality. The chemical oxygen demand graph showed an increasing trend and then it decreased and lastly increased again. So the average COD obtained was 53.5 mg per liter, which classified the river as class 4 based on NWQS. The COD has no change as the data obtained previously is almost the same as the data obtained now. The total suspended solids graph showed a steady trend initially, and then it experienced a steep drop and later on a steep increase. TSS had an average value of 19.89 mg per liter and it was classified as class 3 based on NWQS. TSS value showed a decrement compared to the values previously obtained. Lastly, the ammoniacal nitrogen graph showed a steady trend towards the end. An average of 4.29 mg per liter per station was obtained. Therefore, it classified the river as Class 5 based on NWQS. There is a slight decrement in the AN concentration after the usage of effective microorganisms. Therefore, the usage of effective microorganisms does make a difference in the content of pollution in the river water.
Now, moving on to why we consider EM mud balls as particularly striking. Hi, my name is Nisha and here I am to elaborate. Did you know that EM mud balls, which uses the concept of environmental biotechnology, is being implemented in over 130 countries all over the globe? If you didn't know then, well now you do. So now we shall see why EM mud balls are considered to be a fascinating idea. First is because of its small size and simple technology where it comes in the size of a tennis ball, and second, it is 100% organic matter where it uses molasses, sugarcane and dry cane. Now we shall see the previous implementations of these mud balls throughout the years. First is in Gurnesh Drive in 2009 where the sludge has been reduced to up to 10 cm, and second is in Klang River in 2017 where 2000 mud balls were tossed, and third is in Damansara River where the Management and Science University students have tossed around 178,900 mud balls in their river revival project. Although this research is interesting, it is important to note that this research has its gaps, which will be addressed by Shamirah.
Now, let's move to the sort of area or question that is not answered within this study, which is also known as research gap. Firstly, we found that from this study, it fails to expound on the sustainability of water since the pollution continuously flows into the river and the mud balls are not regularly being thrown. Perhaps the river might be polluted again. Secondly, from this study, the effective microorganisms, also known as EM, are effective to reduce the ammoniacal nitrogen, BOD, COD, and temperature. Perhaps it is not effective to reduce the pH, dissolved oxygen, and total suspended solid. Therefore, from this study, it fails to explain regarding the effectiveness of bacterial additive used. Moving to next, here are the approaches that we had proposed to the research gap. One of the approaches are the System Dynamic Simulation, SDS. SDS is a tool that helps description of system structure. This model is crucial for water resourcing planning to evaluate the sustainability of water resources system in Malaysia. In short, the systematic monitoring could provide a useful information about effective microorganism-based water quality improvements. Besides, the sustainability issues of water supply also could be addressed. On the other hand, a regular monitoring process is also required, for instance, sampling a sample per week in order to determine the changes between application of effective microorganism. This would allow observation and monitoring similarities in condition between these septic tanks of water, which impacts can occur through the significant changes in temperature and also pH. That's all from us. Thank you.